Hello, everybody. How are you? Here I am, Teacher Malafkas Samangistu. I am coming with another topic, which is topic, of course, one remaining topic from virology, or the section that we have discussed about viruses. Viruses. Here I come to the economic importance of viruses, or simply reasons for studying viruses. We shall see some points why humanity studies viruses. It's part of virology. Please focus on the economic. This is simply a reason for studying viruses means simply the economic advantage, economic importance, not advantage, economic importance of viruses, which may include the economic importance includes the negative as well as the positive aspect of virus. And here we come. Some viruses cause disease. This is a negative aspect, of course. Some viruses cause disease in humans and also other organisms. Of course, viruses are important agents of many human diseases, ranging from the trivial common cold to the lethal one, rabies, and of course you can mention many others like HIV/AIDS, the recently historical pandemic that is worldwide spread virus that is coronavirus. Even there are re-emerging viruses. One big problem as far as human health is concerned is emerging and re-emerging viruses, of course. Therefore, whenever we are talking about negative economic importance, we raise viruses as pathogens causing viral diseases, some deadly diseases. Then viruses also play roles in the development of several types of cancer. There are some carcinogenic viruses, including the HIV/AIDS. HIV/AIDS itself can cause a type of cancer, the skin cancer, which is known as Kaposi sarcoma. There are some others now. For example, human papilloma virus. HPV, a human papilloma virus, is well known in causing cervical cancer and reproductive cancer in females, including even vaginal cancer and uterine cancer, most commonly known in causing cervical cancer in females. The second, studying and knowing viruses permits the development of effective means for prevention, diagnosis, and treatment of virus disease. Of course, this is a positive economic. When we know viruses, we can effectively fight viruses again. It's very important. The knowledge of virology is very important for designing drugs against viruses and also designing vaccines against viruses, of course. Therefore, for diagnosis, treatment, and designing drugs and vaccines, we should know them, viruses. Uh, the other, veterinary virology and plant virology. Veterinary virology means as far as our domestic animals are concerned, or animals in general concerned. And plant virology are also important because of the economic impact of the many viruses that cause disease in domestic animals, and plant viruses causing destruction in our crop plants. Therefore, we have to. This is the negative aspect of the economic importance of virus, of course, because viruses destroy our crop plants. There are plant viruses, especially RNA viruses. RNA viruses are well known in plant pathology as far as virology is concerned. Veterinary virology, all groups, the DNA, the RNA, and are also known in causing disease in our domestic animals. Uh, another area where viruses can cause economic damage is in the dairy industry, where the bacteria, the viruses which destroy our important bacteria like lactic acid bacteria, can infect the lactic acid bacteria that are responsible for the fermentation that produces cheese, yogurt, other milk products. You see, the beneficial bacteria are destroyed by these enemies of bacteria known as phages or bacteriophages. Uh, some viruses are useful aspect. Some viruses are studied because they have useful current or potential application or future application. For example, one, phage typing of bacteria. That means some groups of bacteria, such as Salmonella species, are classified into strains on the basis of the spectrum of phage to which they are susceptible. And this is known as phage typing. You see, it is used to classify a species of bacteria into strains, subspecies, strains, based on which phage is infecting which subspecies, which subspecies or group of bacteria. Therefore, this is known as phage typing of bacteria. We can use the phage in order to classify species of bacteria into strains or subspecies, subspecies, if you like. The other is identification of the phage type of bacterial isolates can provide useful epidemiological information during outbreaks of disease caused by this bacteria. This is information related to the previous slide, of course. Yes, when we are phage typing, when we are identifying bacteria based on the phage type they are susceptible to, it can provide useful epidemiological information. How that it is spread in a community and all related aspects of the epidemiology of that disease can be easily studied in a community. This is the advantage. And the other source of enzymes, many viruses are used as source of enzyme. A number of enzymes used in molecular biology are virus enzymes. Example include reverse transcriptase from retroviruses and RNA polymerase from phage have been derived, you see. The other advantage is we can use them as pesticides. Viruses have been used as pesticides. Some insect pests are controlled with baculoviruses. Remember, baculoviruses, myxoma viruses has been used in control of rabbit. Myxoma virus to control rabbit number. Rabbit highly populated. When rabbits reproduce, rabbits have been introduced in Australia one time when the environment was very conducive, so that rabbit population overpopulated the environment and caused environmental destruction, so that to destroy those rabbits and control their number, scientists introduced myxoma virus, highly effective, infectious to rabbits, and destroying the virus, the rabbits by using these viruses. Therefore, you can use this to control rabbits, population of a certain animal if it is beyond that carrying capacity of the environment. Otherwise, baculoviruses are used to destroy pests. They are very important biological control instruments. The other antibacterial agents, we can also destroy some negative bacteria or the harmful bacteria by using phages, bacteriophages. In the mid-20th century, phages were used to treat some bacterial infections of humans. Interest waned with the discovery of antibiotics, or for when antibiotics come to control bacterial diseases, the interest in using phages or bacteriophages as instrument to destroy infectious bacteria is declined with the discovery of antibiotic, but has been renewed with the emergence of antibiotic resistant strains of bacteria. You see, now the problem is antibiotic resistance. Many bacteria are developing resistance to many of our antibiotic drugs, even the broad spectrum antibiotic drugs. So now researchers are picking their studies from where they have thought about phage therapy. That means using bacteriophages to destroy infectious pathogenic bacteria inside the human body or animal body, of course, using this is known as phage therapy. Anti-cancer agents, viruses are also used. Genetically modified strains of viruses such as herpes simplex virus and vaccinia virus are being investigated for treatment of cancers. These strains have been modified so that they are able to infect and destroy specific tumor cells without harming the normal cells. Therefore, there is huge research in the cancer research. One area of research is researching using viruses to destroy cancer cells inside the living body. Therefore, there is hope, of course, anti-cancer agent can be, viruses can be used as anti-cancer agent as far as their positive economic advantage involved. And the other gene vectors for protein production. You see, we can use them. Viruses are used as vectors to transfer the gene that we desire to transfer into another organism in the process of genetic engineering. Therefore, gene vectors for protein, viruses are used as gene vectors for protein production or to insert specific genes into a living cell or tissue. This is practical, of course. Viruses such as certain baculoviruses, adenoviruses are used as vectors to take genes into animal cells growing in culture so that the gene product will be produced. This technology can be used to insert into cells genes encoding useful proteins such as vaccine components, and the cells can then be used for mass production of the protein. You see, you introduce the gene into living cells by using the viruses, which serve as vectors to transfer the gene, and the cell that has received the gene will produce the gene product, that is protein. That protein could be vaccine, or any hormone, could be hormone, or it could be enzyme. And this is when we are using viruses as vectors to transmit or transfer genes of desired type into living cells. And the other gene vector for treatment of genetic diseases, this is a redundancy, of course. Children with severe combined immuno gene vector for treatment of genetic disease. Of course, this is another area of genetic engineering. We are using viruses as vectors now to transfer normal genes so that we can, by we transfer normal genes in individuals with genetic defect, genetic disease, to treat genetic diseases. Children with severe combined immunodeficiency, which is known as baby in the bubble syndrome, they call it, have been successfully treated using retroviruses as vectors to introduce into their stem cells a non-mutated copy of the mutated gene responsible for the disease. You see, the mutated gene is the gene that has been changed so that causes severe combined immunodeficiency, which is severe immune deficiency. These babies are unable to fight even the slightest disease. They can collapse even with the weakest virus or bacteria. They are weak, compromised immune system, genetically born with severely compromised immune system. So to treat with genes, normal genes, replacing the mutated genes, we can introduce normal genes into this individual so that we can cure them. This is very important. And the other is viruses. Virus studies have contributed to knowledge, much of the basic knowledge of molecular biology. You see, the study of virus has contributed to the growth and development of the knowledge of molecular biology, of course, especially, and genetics. Much of the basic knowledge of molecular biology, cell biology, and cancer has been derived from studies with viruses, you see. Therefore, they do have contribution. They have a role in increasing our knowledge in these areas like molecular biology, cell biology, cancer, genetics. I'm winding up this section. Thank you very much. I will come. Please don't forget subscribing, sharing, and liking my videos. Thank you very much. We will come. I will come with another lesson. Thank you.